Alright, looking at part 2 of the cells, we're going to get into the structure of it today. Taking a look at what our objectives were from the last one was identifying the different methods used to identify parts of a cell. That gets into the microscopy, how we're able to separate out cells, cell structure, how we can take a look at them. Remember, please look in your book on page 49, talking about the different types of microscopy, like cell differentiation, centrifugation, as well as fluorescent microscopy. We also compared and contrasted prokaryotic versus eukaryotic cells. What at least three things that all cells are going to have in common, that being the ribosome, cell membrane, and that is cytoplasm. But in a eukaryotic cell, you have other internal bound organelles, starting at the nucleus and then working your way out. Identify organelles that are vital to eukaryotic functions. So listing all the different organelles. In the last video, we just looked at the different membrane-bound organelles. That would be things like the nucleus, which we'll see a little bit more here in this one. But the ERs, the Golgi body, vesicles, vacuoles, peroxisomes. Okay. Identify the three parts of the cell theory. Schleiden, Schwann, and Virchow had developed it, talking about cells are the smallest living thing. If you're alive, you must be made up of those cells, and they're coming from other cells. And then explain why, in terms of the efficiency, cells are so small. So, looking at part 2, the cell, we're going to look at some of the genetic material and what there comes into play, starting out first with the nucleus and the ribosomes. Nucleus's function is to hold on to our genetic library. Now, as I mentioned to you before, if I look at just chromosome number 1, we've got 2 pairs of each set. We are considered diploid. Chromosome number 1, if there's 2 pairs, just chromosome number 1 from mom, 295 million A's to T's, C's to G's. So when we're looking at that amount, there's a lot of genetic materials, a lot of books in that library that they need to go through, and we need to go through and identify all the structures. Okay. Some genes are also going to be located inside the mitochondria and chloroplast. Now, some of those genes are actually going to be coded. Mitochondria have their own DNA, so do chloroplasts. Now, one of the common misconceptions is that mitochondria are only in animals, chloroplasts are only in plants. Mitochondria are in us, but plants have not only chloroplasts, but they also have mitochondria as well. So if I look at the nuclear structure, the structure is separated out by a cytoplasm, by the nuclear envelope, nuclear membrane. So it does have a phospholipid bilayer that separates out the nucleus from the cytoplasm. It has pores in it that allows it to pass materials into and out of it, so it allows large macromolecules and particles to pass through with those pores. Remember what type of molecules need to be able to pass through are things like your mRNA, your structures that need to be able to go through, your ribosome, your rRNA have to be able to go through. So larger structures, so we have to have some sort of a protein embedded within it. So here's an illustration that shows that. We have our nucleolus, which is densely packed rRNA. That's the creation of the ribosomes that are going to be located within there. All the little strings that are thin around it is the chromatin, the genetic material. So all of those millions of books that contain our genes. Now, those genes are going to have to be transcribed, written in, those books have to be written into a different language, which is we call it our amino acid language, or mRNA, and then they're translated by the ribosome at this process out here on the ER, and the ribosomes are then going to help assemble by creating monomers of amino acids and putting those together into dipeptides and polypeptides, which then is synonymous with your protein. So looking at that process, how it takes place, your DNA. This is going to be an entire another test, so it's nothing you have to really worry about here. We're looking at our DNA strand, how it creates the mRNA process and the process of transcription. We are going to remove some of the excess material called introns. Now, this is wasted material, things that we don't actually utilize. So we cut out those pages of the books. It's kind of like if you're writing a book and you have all sorts of material in there that you just wanted to have as filler. So if you needed to write an 8-page paper, you only wrote enough to make 4 pages. You had 4 pages of fluff. Well, we're just getting rid of that fluff. We're just going to get back down to the main pages, things that are needed. You get this. That is now mature mRNA. It's going to leave and it's going to find the ribosome. And at the ribosome, it's there to help look in. It's going to select and read the structure, and it's going to assemble the amino acid sequence together. So looking at the DNA structure, the nucleus structure, it's within the nucleus, all the genetic material. So we have the nucleolus, that's where the rRNA is at, but then we have all of our chromosomes, which is our genetic material that's within there. Okay. Nucleus structure, it's densely stained region, so it's one of the things that we should be able to see with our compound light microscopes. We have no problem finding it. It just looks more like a purple structure. Ribosomes, what they have, they actually have two subunits. They have a larger subunit and a smaller subunit. And they have what they call the A, P, and E site, where the mRNA sequence is going to run right through the middle part of it. Think of it like a top and a bottom of a hamburger bun. As the top and the bottom of the hamburger bun goes through, mRNA sequences through it, and we then are able to put the amino acids together. Okay. Now we have two different really types of ribosomes, some that are bound, which means that they are attached to the ER, and some that are free-floating. So they're just suspended. Now, if they are free-floating, the materials that they create, the enzymes or the proteins that they put together, are going to be utilized in the cytoplasm or the cytosol. If they are bound in the ER, so any protein that has been assembled on the ER is going to find its way either embedded in the cell membrane or exported out of the cell. Pro and eukaryotes both have them, different size subunits, but different proteins. Ultimately, looking at what the purpose behind them is for the same functions of creating enzymes to work things out. Okay, looking at the overall structures of a pro versus eukaryotic. Okay, very, very similar. Looking at a couple other structures that have their own DNA that are very unique, those being the mitochondria and the chloroplast. These two are going to correspond to kind of one another. Mitochondria is to be able to utilize glucose to form ATP. Chloroplast is to use sunlight to make some ATP, as well as the carbohydrates. Now, as I mentioned at the beginning here, these are only found, mitochondria are only the ones we have. So the reason we eat is to be able to make ATP and get that food down through their bloodstream into the mitochondria in the cells. Plants, they can take the sunlight to make ATP and their food that they need. Now that the food that they need, they transfer these carbohydrates to the mitochondria so they can undergo cellular respiration. So looking at some of the similarities between them, they're both there to help transport and create energy. They both have double lipid bilayers. They both have kind of an internal cytoplasm. We call it the stroma here. We call it the mitochondrial matrix in there. They are semi-autonomous, which means they can move, they can divide. And what I mean by autonomous is like they can do whatever they want to on their own. They have their own DNA, they can move around, they do what they need to do. Looking at this structure here, so there's a mitochondrial matrix. Inside there in the cellular respiration process is where the Krebs cycle is going to take place. That's where we're going to create a lot of our electrons that are going to be used in the production of creating ATP. But its main function is our powerhouse. It's helping to break down sugars into smaller subunits, and then we can create ATP from that. Now, a byproduct, other thing that it can do is, in the last chapter we talked about, is apoptosis, helping with programmed cell death to destroy cells that are kind of going out of control. Unfortunately, cancer has found its way around it and kind of blocked mitochondria's ability to do that. So if we can somehow get that back on, hopefully maybe one of you guys someday have now found the cure of cancer. Okay, looking at the structure, we have two membranes. We have an outer membrane that's around the outside, and then we have an inner fold membrane called the cristae. That's where another major portion of the cellular respiration process is going to be taking place. So make sure you highlight this, the cristae. That's these inner folds that are within here. Okay. Looking at our structure again, it's shown now this mitochondria, materials are going to be passing through, and inside the space is where its job is to create ATP. In Chapter 3, our third test, we're actually going to be looking at mitochondria in particular for quite a while, and that all the process of ATP production will make sense there. So that's what this is all kind of coming to be. We're not going to really spend a lot of time on that, other than the fact that you can see that it's a phospholipid bilayer. Phospholipid bilayer, materials have to pass through, proteins are embedded within it. Okay. Divided mitochondria, who else divides like this? Bacteria. Bacteria undergoes binary fission, separates out, so these guys also do. So these are kind of like almost some of the basics of bacteria. So it's kind of one of the unique things that talks about, well, what maybe mitochondria were on their own at the one point, which helps lead into the endosymbiotic theory. Almost all eukaryotic cells have these mitochondria. There may be one very large mitochondria to hundreds to thousands of individual mitochondria. Number of mitochondria is correlated to what its job is. So, for example, our brain cells that need a lot of energy, or our muscle cells, are going to have a lot more mitochondria in it than the cells that maybe don't use as much energy. Okay. Animal cells, here's just another illustration so you can see where the mitochondria are located. Now we get into the plant cells, we're going to talk about the chloroplasts. Okay, chloroplasts are the plant organelles. They contain amyloplasts, which are there to help store starches. They find them in the roots. The chromoplasts store the pigments, and chloroplasts, these are the main structures that we utilize, and this is going to be found in the leaves. And in the leaves, these chloroplasts contain a green pigment called chlorophyll that is going to reflect, I should say, the green, but it's going to absorb other wavelengths of light, and the wavelengths of light that hit are going to split water molecules. Now the structure, we have two membranes. We have an outer and an inner, just like that in the mitochondria. It's got the fluid-filled space called the stroma, and inside there is, in the green stacks, is where we are going to be creating electrons, and in the stroma is where we are going to be creating our glucose material, our sugar or carbohydrate. Okay, all there to help, again, use sunlight to make energy and food. So, should look somewhat familiar. Phospholipid bilayer has proteins within it. This is going to correspond to, again, to our next chapter we'll talk about. We'll spend more time into that. Function in photosynthesis, generation of ATP, and again, it's semi-autonomous, means that it can divide when it needs to. So if it can divide when it needs to, it looks fairly similar. They do a lot of the same types of things. They have their own circular chromosome, like a plasmid, their own DNA, just like bacteria does. So that gets us into this theory, the endosymbiotic theory. Well, maybe there is one larger cell that engulfed another. As it has engulfed another, it got to a point where it was able to expand. And let's see if it had there. Don't have that. If you can imagine in this picture, if there was a smaller cell, this larger cell would engulf it. Eventually said, well, hey, you create more energy for us. So at that point, we'll work together. And so that's the one engulfing going endo and working together. Symbiotic theory is if one engulfed through phagocytosis and then it continued to work together. That's it for the individual organelles and the structures there. We'll look at the next chapter, the chapter 4 in the cell membrane here in a little bit.